the first thing I want to talk about is how we develop quality stable cell clones. And essentially what we do is we have our specific vector. We transfect cells to get a pool of cells, and from that we develop clones and develop a stable cell line. But there's a lot of pieces to this. So what we do first is look at multiple functional validations. We don't necessarily look at expression first. We look at functional validation. And by doing that, we do dose-dependent receptor activation if there's a known ligand. We look at calcium, cyclic A, IP1, or reporter gene assays, depending upon what's best suited for that target. We optimize for cell density, and before we start all this stuff, we make sure that the parent is negative for the receptor that we're looking for, because if you have a parent, and I'll show this later on in some of our stuff, that's positive for the receptor, you can get a really confusing signal and a confusing information. It's much cleaner if you have a cell line that does not have the receptor that you're targeting. All of our GPCRs are tagged on the N-terminus with a small tag, a flag tag, an 8 amino acid tag. That allows us to be able to monitor whether the receptor is on the surface of the cell. It also allows us to be able to interrogate that receptor with antibodies. We don't have to have a different antibody for each receptor. We can use the same antibody, and we can actually look at internalization of the receptor through that means. We also go through, after we've done all of the, after we've done the cell density optimization, the dose-dependent receptor activation, we also make sure that our sequence by RT-PCR is still as we expect it to be and nothing's changed. We make sure that the assays at the cells have been tested for mycoplasma. We verify that it's negative by 2 methods. We make sure that the cells grow in a reasonable manner, meaning that doubling time needs to be less than 24 hours. We also make sure that those cells are stable under our hands for 2 months with continuous culturing. And we validate that with receptor expression and function. That's actually really key, because that means that those cells are going to be good for whoever wants to use them. They're going to be stable in their hands if they use our protocol. Now, I said before that it's really important to have a functional assessment of activity, and here are some reasons why. So let me share what this is, because I'm going to show this some more. The bottom panel here is a FACS analysis of the receptor using our flag tag antibody. And in this particular clone, the receptor basically is very little expression. This clone, the receptor has much higher expression. As you can see, the fluorescence level goes, it's higher intensity. This is where the parent is, very, very low signal using our antibody. So what happens here is, this is for the dopamine D1 receptor. When you increase receptor expression, you can see the levels on the bottom. We get differences in functional activity. So in clone 4, this top one, we get really good activity with dopamine and with phenaldipam, but here, bromocriptine, which is supposed to be a partial agonist, doesn't seem to work at all. On the right-hand side, where we have very high expression levels, this partial agonist behaves as a full agonist, which isn't what is seen therapeutically. It actually it should be a partial agonist, it's a full agonist. Here, though, this is the sweet spot, clone number 2, where you can see full agonist activity with the full agonist, but partial agonist activity with the partial agonist. So if you want to, what we do is we work with our clients to be able to make sure that we have cell lines that function the way they're interested in for screen. So if somebody's interested in just having the receptor on the surface for looking at binding, you might want a high expressor. You might want something like this. If you're interested in looking at all of the agonist activity, you still might want to look at this, but then go back to the clone number 2 for partial agonist. Or you might say, okay, we'll just shoot for clone number 2 and look for full and partial agonists at the same time. When we select our clones, we don't just select 1 or 2 of the clones. We actually wind up selecting several, like 5 or 6, with different expression levels, so that we can go back into our freezer and pull out some of these clones if somebody is interested in a different profile that they might be the standard one that we give. So it's actually something really important to know. It's something important for you to know if you're developing your own cells, that the receptor expression It's really important, and it's really important to make sure that you control for that and that you look for the pharmacology that you're interested in. Most of our cells, I didn't say this before, most of our cells are either CHO or HEC293. There's times when CHO or HEC293 don't work for the particular receptor because it contains that receptor that we're targeting. And so in that case, we find alternate cells that don't have the receptor. So occasionally you'll see in our catalog that we have different cell lines, and that's the reason why. Here's another example using glucagon. This is a glucagon cell line. And when we developed it, this is the cell line that we developed with the parent and the expression of the glucagon receptor here. But on the left-hand side here is actually what the functional activity looks like. So both this compound and this compound, which should be a partial agonist, behave as full agonists. But this cell line, which is a different clone, was selected for the partial agonism. And again, you see lower expression level, and then you see partial agonist activity with the partial agonist. Over the years, we've developed a lot of GPCRs. So we now have getting above the 450, heading toward 500 different receptor expressing cells. And this can be cells that are human, most of them are human, they can be mouse, rat, different species, different types of receptors, a whole different battery of choices. And we're now starting to develop cells that contain our receptor as well as a sensor for beta-arrestin, which I'll talk about in a bit. I said we have a functional GPCR safety assessment panel. This is our functional panel. We chose this panel based on input from multiple clients who are in the pharma industry saying that these are the ones that they typically need functional assessment for. And so we developed this panel, and we can run this on the fly if somebody is interested in this. We also have examples here of assay development with both recombinant and native cells. That's what's on this slide. The top is a recombinant cell for the histamine receptor, looking at calcium. This is basically the Z prime over several weeks, months of data, and this is actually the EC50s over months of data. And you can actually see that over this campaign, everything stayed very stable. Now, when we do screening for our clients, if we find that it's very, very heavy QC on the screening, and we make sure that those assays perform as they should. If they don't for any reason, we repeat the assay, we maintain information in a log on what the EC50 or IC50 of the different compounds are, and everything needs to behave properly or the assay is repeated. On the bottom of this particular slide, we show data in primary cells, non-transfected cells. This is looking at endogenous DNA PK autophosphorylation. You can see autophosphorylation. And this is DNA PK P53 phosphorylation by taurin and HeLa cells. Both of these are HeLa cells. And this is a CGRP receptor, and its activation and inhibition in SK and MC neuroblastoma cells. This is just a small example, and you can notice this is not GPCR, this is GPCR. So we have multiple different pathways that we can interrogate.